Hello everyone, this is Dr. Anand, Assistant Professor, Department of Bioinformatics, Vijnan University, Guntur. So yesterday we are all discussed about good laboratory practices. So once I want to rewind the good laboratory practice class. So good laboratory practices means doing scientific work in any research laboratory, so in a clean, organized and healthy manner, so that we will get results are reliable and trustworthy. So as I discussed in the last class, good laboratory practices I divided into 5 different steps. So one is following rules, and whatever we do in the laboratory, so we have to follow the specific standard protocol method and we have to perform the experiment. And second one is keeping clean equipment. So whatever we do experiments in the laboratory, so we have to use the clean instrument and keep instrument work properly. And also we have to use the keeping records. So whatever we do in the laboratory experiment protocol, so we have to write down in the documentation. So we have to maintain the logbook also and documentation purpose. And we have to be safe. So whatever we do experiments in the laboratory, so we have to perform and we have to protect ourselves and others and environment. And also finally we have to be honest, because we should not change the results or make up the data. So if you follow all these 5 good laboratory practices while we are entering into the lab, so some other scientific community want to repeat that experiment. So if you follow all these 5 steps, so that we will mention all the protocol and methods and GLP practice and guidelines, so that whoever want to perform the same experiment in other community, some other place, so they will follow our protocol and they will do the same experimentation, so that they will get same results, so all results will be reproducible. And also I discussed good laboratory practices are what you are going to do in our research laboratory. So this is called good laboratory practices. So as earlier I said, standard definition like laboratory studies are planned, performed, monitored, recorded and achieved. So these all steps we have to follow and we have to do while performing experiment in the laboratory. So like as I discussed I discussed planned, performed, monitored, recorded, reported and achieved. So and also whenever we do any experiment in the laboratory, so we have to wear the appropriate protection equipment, like I said, gloves, apron, these and all. And also whatever we do experiment in the protocol, we have to read thoroughly before going to starting the experiment. And also we have to label your materials, whatever you are going to handle. And also we have to check the label on the container, so before taking out any substance. So next one, so as I said earlier, and next one, these are the safety equipments. So whatever we do experiment in the laboratory, so we have to know the location and how to operate the safety equipments. So then next one, and we have to use the chemical who for the strong acid and other chemicals and solvents. So these all the theoretical about the GLP practices as we discussed in the early class. So today we are going to start about use the recommended biosafety level containment while handling microorganisms. So as I said earlier, so whenever we want to work in a biological laboratory, so we have to use the recommended biosafety containment while handling the microorganism. So today I am going to discuss about biosafety levels. So here you see the picture, biosafety levels, biosafety level 1, 2, 3, 4. So the image shows arrow mark up to down. So these are the risk of microorganism, low level to high level. Suppose for example BSL level 1, so and next 2 and 3 and 4. So the level shows the low risk microorganism versus highest risk of microorganism. So now we will discuss about what is biosafety level in particular laboratory. So biosafety level is used to identify the protective measures needed in the laboratory to protect workers and environment and the public. So whenever we do any laboratory work in the research laboratories, so while you are handling microorganism, we have to protect ourselves and others and environment. So we have to take the safety measures. So So the biosafety level, it will indicates the how we have to identify the protective measures while we are handling different types of microorganisms. And also whatever we do activities or projects or any different works in the biological laboratory, so these are all characterized by biosafety levels. So as I said, mentioned, biosafety levels are 4 types. One is BSL 1, 2, 3 and 4. So BSL 1 is the lowest risk microorganism and BSL 4 is the highest maximum level of containment. So let us understand what are the different biosafety levels. So according to the biosafety level, you have to handle the particular organism. So biosafety level 1, so we are going to handle one type of particular organism, second and another type of microorganism. Likewise, biosafety level 1 to 4, so we are going to handle different types of microorganism as per the risk of the microorganism. So there are 4 biosafety levels, we are all know. So these are 4 types of biosafety levels are classified based on the low risk microorganism versus high risk microorganism. As I said earlier, BSL 1 low risk microorganism and highest one is the BSL 4. These so biosafety level 4, 3 are based on the classified based on the low risk to highest risk microorganism. So now we are going to discuss about the biosafety level 1. So biosafety level 1 is actually those organisms which does not cause any disease in healthy individuals. So now we are going to discuss about 4 different types of biosafety level. So now we are discussing about biosafety level 1. So whenever we want to work in the biosafety level laboratories, so first of all we have to know what type of organism we are going to handle in this type of laboratory and what are the risks associated when we are handling this type of microorganism, and also how we have to take care and what are the precautions, like what lab rules we have to follow while we are doing different types of biosafety levels. Now biosafety level 1, as I said, these are low risk microorganisms we are going to handle. So here for example biosafety level 1 we are discussing, so example is E. coli. So these E. coli is we are routinely using this microorganism in the laboratory various purpose. So here minimum potential hazard to the laboratory personnel and environment. So classical example is E. coli. So what are the lab rules we have to follow when we are handling this low risk of microorganism? So we have to wear the apron, we have to wear the gloves, and we have to wash the hands, and also so these the laboratory rules we have to follow when we are handling the biosafety level 1 low risk microorganism. So when you are handling E. coli biosafety level 1 low risk microorganism, we won't cause, these organisms won't cause any disease in the healthy individuals, but sometimes if you are immunocompromised or if you are having any kind of other diseases, then sometimes you may cause some other diseases, but those are not life threatening diseases. So only low risk microorganisms it may cause. So here if you are healthy individuals, if you are handling E. coli low risk microorganism, it does not cause any diseases, but if you are immunocompromised or if you are handling other kind of diseases, then we have to take care about ourselves while you are handling E. coli. So next one, BSL 1, so we completed. Now we are going to discuss about biosafety level 2. So biosafety level 2 is the moderate potential hazard to the laboratory personnel and environment. So as I discussed, biosafety level is basic level. Now we are discussing about biosafety level 2, moderate risk of microorganism we are going to handle in this type of BSL 2 laboratory. And biosafety level 1 and 2 are the general basic laboratories we are routinely used in the laboratories. So difficult to contact via aerosol in the lab setting. So here one of the classical example is Salmonella, Streptococcus aureus, hepatitis A, B, C, influenza and Lyme disease. So these are the different diseases we may get while we are handling biosafety level 2 microorganism. So what are the precautions we have to take while we are handling biosafety level 2? So likewise, as I said, BSL 1, BSL 2 also we have to wear wear apron and gloves, and here sometimes we have to wear the face shield also, and we have to hand wash. So these are the lab rules we have to follow. We have to follow the precautions when we are performing. So handling the biosafety level 1 and 2 while handling low risk microorganism. So now we are discussed about biosafety level 2, and now we are handling biosafety level 3. So biosafety level, as I discussed, biosafety level 1 and 2 are the basic laboratories. So no need to worry about much while we are handling biosafety level 1 and 2, because those are basic laboratories. Routinely we are using the any experimental purpose in the biological lab. And now we are discussing about biosafety level 3. So here serious diseases. So these organisms when we are handling, so we may cause serious diseases like HIV, H1N1 flu, tuberculosis, SARC, rabies, West Nile viruses. These are the examples for the BSL-3. So when you are handling BSL-3 microorganism like serious causing diseases, so you need proper training, then only you have to handle BSL-3 organisms and as well as the BSL-4 organisms. So they also get spread through aerosol. That's why they are very, very infectious, and they do not have to protect yourself from those microorganisms. The classical example is HIV H1. So here one more classical example is tuberculosis, Mycobacterium tuberculosis. So here whenever you handling BSL-3 serious risk microorganism, so what are the rules follow we have to in the laboratory? So this lab is isolated, and we have to wear the mask, and we have to hand wash. So controlled air flow system is required when we are working with the BSL-3 microorganism. So these are the precautions we have to take care while we are performing laboratory experiment in the lab. So while handling biosafety level 3 organisms, serious disease causing organisms. So example Mycobacterium tuberculosis. And next one, final one, BSL-4. So BSL-4 is the highest risk of microorganism we are going to handle in the laboratory. So here example is like Ebola virus example, classical example. So these are the life-threatening causing microorganism. So if you are infected with BSL-4 organism, these are causing very dangerous diseases, and the exotic posing high risk of aerosol transmitted infections. So whenever we are handling BSL-4 organisms, we may get infected through aerosol transmitted infections. So BSL-4 is the category of microorganism where actually cause the fatal disease, and they are actually life threatening. So as I said, classical example is Ebola. So BSL-3 and 4, these are serious causing organism handling. So whenever we want to work with BSL-3 and 4, so we need proper specific training, then only we have to handle the BSL-3 and 4 type of microorganisms. What are the precautions while we are working on with BSL-4? So we need full suit top to bottom with own air required, and also whatever that lab should be isolated from the other labs. So like separate building we have to maintain. So and also whatever material you are discarding after doing experiment, all the materials should be autoclaved. Like BSL-3 also, here serious organism we are going to handle, right? There also we have to do decontaminate whatever waste you are generating from your experiment, and BSL-4 also whatever you are going to generate the waste, so all the waste should be autoclaved or sterilized, decontaminate. Then only it is good, because if you don't want to decontaminate, if you are not going to decontaminate directly, if you are disposing directly that infectious material into some other dustbin, so it is causing infectious to some other people. So make sure we have to decontaminate whatever waste material generated from your experimental experiment. So these are the different BSL 1, 2, 3 and 3. So here the picture shows how we are performing the laboratory, we are handling the microorganism. So as I said, these are the BSL 1 to 4, these are the low level risk microorganism to high level risk microorganisms. So, and guidelines and precautions and recommended actions. So while we are handling this type of microorganism handling, so what we have to do is decontaminate your workbench and the equipments handling after use, as I mentioned earlier, and decontaminate all the infectious material before disposing them off, and any spillage, accident or injury should immediately be reported to your instructor. So first one here, decontaminate your workbench and the equipment handle after usage. So whenever you are working with these type of microorganism handling in the laboratory, you have to decontaminate your workbench and also whatever instrument we are going to use while handling microorganism, all instruments should be decontaminate, like pipettes you have to decontaminate, and decontaminate all the infectious materials before in disposing them off. Like BSLL 3 and 4 we are going to generate waste material. So because those are infection materials, whatever you are going to generate the waste material in BSLL 3 and 4, because those are serious diseases you may get. So we have to decontaminate all the waste generated from the BSLL 3 and 4, so that all will be safe. There is safe and non-infectious if you are decontaminating all the waste material. And any spillage, accident or injury should immediately be reported to your instructor. So whenever you are handling any microorganism in the research laboratory, so if sometime it may happen spillage of the infectious material, or maybe any sudden accident, or maybe injury to your hand or leg, so if anything happened, this type of anything happened suddenly, so immediately you have to report the laboratory instructor and supervisor, so that he know how to handle the situation. He will guide you proper way, so that we can take care ourselves and other people and environment. So next one. So as I said earlier, so whenever you are going to handle different types of microorganisms in the laboratory, so we have to decontaminate all the waste material, right? So how we have to decontaminate our waste material? So I am going to explain in 3 different methods. So these are the guidelines and precautions and recommended actions. Decontaminate all the infectious materials before disposing them off. So first one is sanitation. So sanitation means surface cleaned and microbial role reduced. Sanitation means, so this is the surface cleaning procedure. So here killing germs, so we can reduce the load of the microbial load. So this is the common solution for the routine cleaning. So a mild cleaning agent such as soap or household detergent will clean the greasy surfaces but may have little antimicrobial activity. So when we are going to perform biosafety level 1 to, we are going to use sanitation, because whenever we are performing any work in the laboratory, so we have to clean the surface. So that time we have to use the cleaning agent like soaps and detergents we have to use, and we have to clean the surface of your workbench, so that all germs whatever present in the germs we can kill, and we can reduce to the microbial to safe level. So next one, disinfection. So this is the second method. So disinfection, so microbes on surface killed, inactivated and made harmless. So this is the second method. This is also we can use when we are using biosafety level 1 and 2 handling microorganism. So this is also the solution for simple cases of contamination. So use quaternary ammonium compounds which have antimicrobial properties. They do not corrode metal or damage products. So when you are want to perform the disinfectant in your area, so you can use quaternary ammonium compound and also alcohol 750 and also phenol and bleach also we can use. So these solution, it will clean the workbench, so that kill the germs on the surface of the workbench. So this is the disinfection second method. So first one sanitation and second one is disinfection. These 2 methods we can perform while we are handling the biosafety level 1 and 2. And coming to sterilization, so the sterilization protocol when we have to use, so while we are handling biosafety level 3 and 4, because those are we are handling serious microorganisms, right? So what is sterilization? Like steam sterilization, so it kills all the life. kill the all life of the germs. So total removal of any contamination, no viable microbes remain. So when we are performing the sterilization, so it kills the all forms of lives. So removal of any contamination. Reserved for the serious contamination, as I said, BSLL 3 and 4. So we know that serious contamination will be there. So waste material will be generated. So we have to use at the time sterilization autoclave. Like use oxidative agents such as bleaches, hydrogen peroxide, stick to the safety rules and don't use these agents on metal or plastic. So sterilization, we are going to use autoclave like steam sterilization and also radiation and also dry heat. These tools we can, these instruments we can use if you want to clean the that area using sterilization while you are performing the BSLL 3 and 4. So these are the three different methods if you want to decontaminate waste material generated from the while you are handling different types of microorganisms when you want to perform in the research laboratory. So these are the different results. So finally, summary of our good laboratory practices. In first class, so I have discussed about good laboratory practices. So what we have to follow in the laboratory whenever you want to enter into the lab, when you start off the experiment. So as I said earlier, so whenever you want to perform laboratory experiment in the protocol, so we have to know the theoretical study about that experiment and what are the chemicals, what are the solvent you are going to handle, and also whenever you perform any chemical reactions, we have to know what are the byproducts generated from your experiment, so that we have to careful and we have to take precautions. So whenever you want to perform any laboratory work in the research laboratory, so we have to know the theoretical study and also we have to use proper instrument and clean instrument, working instrument, so that we will get the proper results. And also as I said earlier, we have to use the proper documentation. So we have to write the total procedure step by step, first step to last step. We have to write all the procedures so that you will perform each and every step, so that if someone want to repeat the same experiment, scientific community, so they know what is our research protocol, we will share whenever they required, so that they will also follow the same protocol method, they will also get the same results. So in research laboratory, our biological lab, whenever you do any research work, so we have to maintain the lab record and we have to follow the lab rules and regulations, step by step standard method we have to follow. And scientific community want to repeat our same experiment, if they want to replicate the experiment, so we know the step by step procedure of our experiment, so we will share and they will also follow the same procedure, and also they will get also same result as reproducible. So we have to follow the proper scientific procedure when we are performing the research work in the research laboratories. So we know good laboratory practices. So while you are handling different types of microorganisms, we know the precautions and good laboratory practices. So we know what to do, what not to do, and also what are the recommended actions, what are the not recommended actions. So data register, when we are performing an experiment, we have to maintain the data is important and results is important and proper documentation is important. So likewise in good laboratory practices, so as per US FDA protocol, so we have to follow the good laboratory practices like rules and regulations. So that while keeping in mind when we are following the rules and regulations of GLP practices, so that while we are performing under the preview of GLP practices, so that we will get the good results, and also other scientific community, if they want to repeat our same experiment, so they will also get the same results, so that the results are reproducible. Finally, the results are going to help to the human society. Thank you, one and all.